Good morning, everyone. My name is Emily. I recently defended my PhD in Dr. Dorota Skronster-Kravchik's lab, and we're interested in understanding the molecular mechanisms driving age-related visual decline. And so when we think about aging, we like to think of it as three interconnected gears. The early molecular changes that occur before any overt phenotypes, the compensatory mechanisms that cells and tissues deploy in order to maintain function, and then the resulting aging phenotypes that emerge as a result of insufficient compensation. And using this model, we can take a closer look at the molecular changes that happen before any overt dysfunction. And of these molecular changes, epigenetic regulation has emerged as one of the key players in aging. And epigenetics refers to changes in the DNA without actually affecting the underlying sequence. And so within the epigenetic modifications, DNA methylation has been shown to be a well-characterized and well-studied phenotype in aging. And this involves the addition of cytosine groups the addition of methyl groups to cytosine residues in the DNA. In aging, there's two major patterns of DNA methylation. There's global hypomethylation, where there's loss of methyl groups throughout the genome, and this can result in genomic instability and inefficient gene oppression. But on the opposite, on the other hand, in specific sites such as CPG islands and gene promoters, there's actually hypermethylation, and this has the opposite effect. So now there's loss of expression control, and there's inappropriate silencing of genes that should normally be expressed. And importantly, so a lot of these DNA methylation changes are stochastic, but there are certain markers that are actually very well reproduced throughout aging, and we can use these to create these methylation or epigenetic biological clocks that can be used to quantify biological aging. And we can use these clocks to show whether certain exposures or conditions are actually associated with accelerated aging on the DNA methylation level, and whether certain interventions are actually turning back the clock. And so among these DNA methylation markers, there's been a lot of one has significantly emerged as a key epigenetic marker of aging, and this is in the E-level2 gene. And so in 2012, these four CPGs in the E-level2 gene were identified as being significantly correlated with aging. And this has since been shown to occur across several tissues in the body, some of which I'm highlighting here in the brain, the liver, and the thyroid. And so recently in our publication, we also showed that there were two variants in the human E-level2 gene that were significantly correlated with an accelerated or increased progression to intermediate staged AMD. And so while E-level2 has been widely used as a biomarker of aging, it actually encodes a functional enzyme in the fatty acid elongation pathway. Specifically, it catalyzes the first and rate-limiting step of fatty acid elongation, where there's the two carbons are added to an existing fatty acyl chain. And so E-level2 is important for the production of both omega-3 and omega-6 long chain and very long chain, or LC and VLC PUFAs, or polyunsaturated fatty acids, which contain more than two double bonds in their structure. Specifically, it sits at a critical metabolic branch point because the product of E-level2, 24,5 omega-3, can either be further processed in the peroxisome to produce DHA, or docosahexaenoic acid, 22,6, or 24,5 can also be further elongated by other enzymes such as E-level4 to the very long chain, or VLC PUFAs, which are important in the retina. And so, as I alluded to previously, these polyunsaturated fatty acids are really important for membrane properties and really dictate the biophysical properties of the membranes that the PUFAs are incorporated into. And so largely they're incorporated into complex lipids such as phospholipids, which make up the lipid bilayer, and the saturation and the chain length it dictates the properties such as membrane fluidity, flexibility, the thickness, as well as interacting with membrane proteins themselves to affect their function. And there's been a great body of research in the literature showing the importance of PUFAs in the retina, starting from development. DHA is critical for both brain and retinal development. And omega-3 rich diets have also been shown to be neuroprotective in several retinal diseases. And also in human donor eyes, there was decreased levels of LC and VLC PUFAs in aged human donors, which was further exacerbated in donor eyes with AMD. And epidemiologically, diets rich in fish, which have a lot of omega-3 PUFAs, have been shown to be protective against the progression to late stage of AMD. However, results from large clinical studies, such as the AREDS2, did not show any additional benefit with the addition of DHA and EPA. And so it's important to really understand the metabolism as well as the regulation of how these long-chain PUFAs are produced, and how these changes in aging can impact retinal membranes and overall function. And so knowing that the RPE serves as the metabolic gatekeeper to the photoreceptors, also is the site of primary damage in AMD, we decided to take a closer look at polyunsaturated fatty acid metabolism in the RPE. And so we know that it's a two-way relationship between the RPE and the photoreceptors. The RPE forms the outer blood retinal barrier, delivers fatty acids from the blood, secretes growth factors to support the health of the photoreceptors, and also recycles lipids through phagocytosis. And on the opposite direction, photoreceptors secrete lactate and then also deliver this large bolus of proteins and lipids to the RPE that needs to be processed via phagocytosis. So what we first did was we looked at a previously published data set where they looked at healthy human donors across several ages and performed RNA sequencing from the RPE from these donors, and then using the genes that they identified as being significantly correlated with age, we wanted to take a closer look at the cellular components that were really impacted on the transcriptomic level in healthy human aging. And so we identified that a lot of these cellular components were largely membrane components, such as the plasma membrane, basement membrane, basolateral plasma membrane. And we also correlated this in our in aged mice. So we compared the transcriptomes of 3 and 18-month-old mice, and similarly performed RNA sequencing, gene ontology analysis of the cellular components, and also identified that many of these membranous components were significantly impacted with aging. On the fatty acid level, we performed fatty acid quantification, and we see a depletion of these long chain and very long chain PUFAs in the aged mouse eye cup. And so to correlate this with direct loss of E level 2 activity, we have we turn to our mutant model. So our E level 2 C234W mutant mice, they lack the ability to convert 22.5 to 24.5. And so we also see this similar trend in this depletion of the long chain and very long chain PUFAs, as well as changes in their overall lipid classes. And then when we take a closer look at the DHA incorporated lipid species, many, if not most of these DHA incorporated lipids are down in the E level 2 mutant eye cups. And not only are the DHA incorporated lipids decreased, but overall just PUFA incorporated lipids are also decreased in the E level 2 deficient eye cups. And so similar to gene ontology, with lipidomics data, you can also perform lipid ontology, which gives you a better idea about kind of the functional and cellular aspects of the lipid species that are significantly changed. And so using the lipid species that we identified as being significantly changed, we performed lipid ontology and found, again, a lot of the membrane components and plasma membrane being significantly impacted, as well as the ceramide and sphingolipid classes. And so taking a closer look at the morphology of the RPE, We performed immunofluorescence on flat mounts dissected from E level 2 mutant mice. You can see in the top, these are wild type Z01 stain. The nice hexagonal structure is maintained. And interestingly, in the E level 2 mutant mice, there's kind of this pulling away and this interesting phenotype in their RPE cells where they lose this nice connection between each other. And we can further quantify that using the reshape pipeline that was developed in Dr. Kapil-Bharti's lab at the NEI. And we see that there's changes in the cell area as well as changes in the aspect ratio in the mutant mice. Taking a closer look, we performed electron microscopy with Dr. Bonilla at Case Western. On the right here is the level 2 mutant mice at a higher magnification here. We see there's loss of basal infoldings as well as an accumulation of this debris in the basolateral surface of between the RPE and Bruch's membrane in the level 2 mutant mice. And then so to further characterize and study the effect of the loss of E level 2 in RPE cells, we turned to an in vitro model of RPE cells. So we followed this protocol that was developed in Dr. David Williams' lab, where we differentiated ARP19 cells using nicotinamide. We performed siRNA-mediated knockdown of E level 2. And then importantly, the day after this knockdown, we switched the media from FBS containing to B27 because FBS contains the products of E level 2. And then after that, 6 days after this, we performed experimental assays. And this is just showing that there's an increase in RP65 expression with this differentiation protocol and still very good functional knockdown or very good knockdown of E level 2 on the gene level. And then functionally, we quantified fatty acids and saw an accumulation of substrates and depletion of the E level 2 products. Again, we performed lipid ontology on the significantly changed lipid species and identified membrane component as being significantly changed plasma membrane, as well as ceramides coming up again. And then actually looking at the individual ceramide species, we see that many of them are also increased on the lipidomic level. And so we see that E level 2 deficiency impacts membrane. So we turned to take a closer look at the plasma membrane specifically. We perform plasma membrane fractionation following centrifugation protocol. This is just a Western blot to show that we have nice enrichment in a plasma membrane marker, the sodium potassium ATPase, and they're relatively clean, noted by the lack of cytosolic, nuclear, or organelle markers in the plasma membrane fraction. And so we took these plasma membrane fractions, and with our collaborators at UCSD and Dr. Itai Budin's lab, we performed C-Larden lipid staining. And C-Larden is a membrane phase-sensitive fluorescent dye that integrates into lipid bilayers, and it changes its fluorescence, its emission based on the lipid environment. And so these are the traces that we had obtained from the control and E level 2 knockdown plasma membrane fractions. And we can quantify this as a ratio by looking at the peaks at 440 and 490 nanometers. And this gives us what's called a generalized polarization ratio, where a higher GP value reflects a more rigid or more ordered plasma membrane. And so in the E level 2 knockdown cells, from this quantification, we see that the plasma membrane order is increased, which signifies a more rigid plasma membrane. And so on the lipidomics level, we also performed lipidomics of these plasma membrane fractions. We see increase in lysophospholipid species, as well as an increase in sphingomyelin, and this increase in ceramides, which again has come up several times on the lipid ontology and lipidomics data before. And to take a closer look, we also perform immunofluorescence of ceramides in these cells, and we can see that there's a striking accumulation of ceramides in the knockdown cells. And using this M-cling or this membrane marker, we can see that a lot of these, the accumulation of ceramides is close to the plasma membrane in the periphery. And so ceramides are known to induce membrane stress. and also because of the shape of the ceramide lipid, it can also drive negative intrinsic curvature, which is this inward budding of the membrane. And so we wanted to assess whether there was membrane stress occurring in these cells. And so one of the ways to look at this is looking at the loss of lipid asymmetry. And so what I mean by that is normally the lipid bilayer is asymmetrically distributed. So lipids such as phosphatidylserine or PS is normally found only on the inner leaflet of the lipid bilayer. However, under plasma membrane stress or injury, this PS is then flipped out to the outer leaflet, and annexin 5 can then bind. And so to first to assess this lipid asymmetry, we stained live cells with this fluorescent annexin 5 probe, and we see that there's increase in annexin 5 binding to PS in the knockdown cells. However, because we also know that annexin 5 binding can also signal apoptosis, we wanted to also address whether we wanted to see if there was apoptosis occurring. And so as a kind of a positive control, we incubated cells with hydrogen peroxide. And then this is the level of annexin 5 binding when the cells are actively undergoing apoptosis, and I'll also show a couple more assays later to show that we don't think that there's apoptosis occurring. We
Emily?
also, yeah.
Sorry, can I ask, are you okay with interruptions?
Yeah, of course.
Okay. So in this Eloval-2 knockdown versus hydrogen peroxide, I mean, hydrogen peroxide is definitely giving the expected kind of membrane localization, but with the Eloval-2 knockdown, the localization of that annexin 5 seems to be like perinuclear, or it's not as clearly plasma membrane. Could it be a different organelle that also carries annexin 5, or like some kind of vesicle? I don't know much about the biology there.
Yeah, good. Yeah, that's a great question. I think, yeah, with this, like, because this is like like a one slice image of a 3D cell, it's hard to say like exactly if the signal is, sitting like in the 3D space of the cell. However, I think because annexin 5 is pretty specifically binding to PS, and I'm not sure if PS is like, say, on like the mitochondrial membrane, like where it is there. I feel like it's also on the inside of the mitochondrial membrane, but I would have to look into that. I
Can I
don't know
just
if
mention something quickly?
yeah.
You're presumably not permeabilizing your cells, right? Otherwise,
No.
all the internal PS would be there. So I don't think you're hitting any organelles with this, right?
Yeah, that is true.
Yeah,
Yeah.
it would
No,
have
these
to
are
be
live
something
cells.
extracellular. Yeah.
Yeah, these are not permeabilized cells.
Yeah, but I was curious too. The blotches are quite interesting that you have these large patches of annexin. It's quite curious.
Hmm.
Are PSs enriched in lipid rafts at all? Or could there be domains that you're really finding that might not normally exist? Or I don't know. Just trying to figure out
Um,
why isn't it sort of evenly spread if it is?
yeah. I know ceramides make up a lot of the lipid rafts. PS,
I wouldn't think so either, but
I
I
wouldn't
think
think so because yeah, a lot of times in the context when PS is flipped out, people are, the Nexin 5 and then apoptosis. Or like,
Can I
for
chip in here?
phagocytosis, like eat me. Mm,
Can I chip in?
yeah.
So I think one thing is, first of all, what was said by Philip, that it's non-permeabilized. But then if you think about it, those are breaks. So if they are all over the plasma membrane, like completely evenly distributed, the cell would die. I don't think it would be possible. So it is localized and we are catching it in a perfect moment. I am also very much intrigued by those big blobs, but with Emily, we think it's because of actually production of ceramides in those places, right? So
Mhm.
there is because well, we will show it a bit later. She will show that there is extracytosis on enzymes. So we think that maybe
Spoiler
explain
alert.
on. Will we? All right. So maybe later she can come back to those patches.
Okay. Can I ask one more follow-up? Have you done any pulse chasing with another color annexin to see how dynamic these are? Like, is it possible the membrane's kind of flipping and flopping or something like that?
No, we
Okay.
haven't. That'd be
It's
great.
probably the case.
Cool. Thanks.
Okay, yeah, so this is an orthogonal assay that we also performed where we can simultaneously monitor PS to an exon 5 binding, as well as any downstream activation of apoptosis by DNA dye uptake. And so again, we see in the gold trace this increase in PS and an exon 5 binding in the E level 2 cells. However, there's no activation of apoptosis. And this is in comparison to a positive control, cells incubated with hydrogen peroxide. So this increase in PS and an exon 5 binding followed by activation of apoptosis. And to really nail this and bring this home, we also performed caspase staining in these cells. So first in the non-treated control and E level 2 knockdown cells, there's no difference in caspase 9 or cleaved caspase 3. And then the levels of these, two proteins when the cells are incubated with hydrogen peroxide. So to recap, we have an accumulation of ceramides. There's membrane stress, loss of lipid asymmetry in the lipid bilayer. However, the cells are not dying. And so this led us to turn to some of the plasma membrane mechanisms that cells employ in order to maintain health and avoid cell death. And so there are many repair mechanisms that cells can use, such as patching, tension reduction, budding, and lysosomal exocytosis, which we focused on. And this lysosomal exocytosis involves these breaks in the membrane, an influx of extracellular calcium, which drives lysosomes to be redistributed peripherally. And these lysosomes can then secrete an enzyme called acid sphingomyelinase, or ASM. And this enzyme cleaves sphingomyelin on the membrane to generate ceramide. And ceramide then drives this negative intrinsic curvature, or this inward budding to remove these lesions. So to investigate whether this mechanism was occurring in the cells, the first thing we did was calcium measurements. And so with Dr. Authy's lab here at UCI, we used a genetically encoded calcium sensor called SALSA6F. There's a stable TD tomato expression followed by a calcium sensitive GCAMP6F. So we can get a ratiometric quantification of calcium signaling in the cells. And so this is just some videos of control and knockdown cells on the bottom showing the dynamic flashes of calcium signaling. And these are cells that are just incubated in normal extracellular calcium conditions, so 2 millimolar calcium. And then with this imaging, we can also obtain the single cell traces of calcium activity and quantify this ratio. So on top in the control cells, showing the single traces, and then on bottom, the E level 2 knockdown cells, you can see these more active spikes in the peaks in the calcium signaling. And then we can actually quantify the cumulative calcium over time by quantifying the area under the curve. And we see there's an increase in calcium in the knockdown cells. And then looking at the average traces of the cells, so again, this is in the normal extracellular calcium conditions, 2 millimolar, and then there's an increase in basal calcium activity in the knockdown cells. And this is lost when we remove calcium from the media. So in zero calcium conditions, this increase is lost. And so from this, we conclude that there's increased calcium activity in the knockdown cells, and the source appears to be extracellular, because when we remove that extracellular source, this increase is lost. So next, we looked at the localization of lysosomes, because this influx of calcium can then trigger the lysosomes to be redistributed. So again, we used mCling as a membrane marker, and we labeled lysosomes with LAMP1. And then we quantified the proportion of lysosomes within the cell from the perinuclear to peripheral region. And in the knockdown cells, we see that there's a redistribution of the lysosomes to the periphery, while in the control cells, they remain in this kind of, 30%, 40% perinuclear cloud. And then to take a dynamic look at the lysosomes with collaborators at the Schoenberg at UCSD, we performed 4D lattice light sheet imaging in live cells labeled with lysotracker to track these lysosomes and their movement over time. And so again, we see in the knockdown cells that there's a redistribution of lysosomes to the periphery. And also these lysosomes that are in the periphery seem to be more dynamic, quantified by their net lysosome displacement, their average lysosome speed, as well as their diffusivity. And so we see that there's a redistribution of lysosomes to the periphery that are more active. And to see exactly how close they are, we can use proximity ligation assay, or PLA, where we incubated cells with lysosome markers, so LAMP2, and then a membrane marker, ZO1. And normally, there's only signal produced if these two antibodies are within 40 nanometers of each other. And it produces this yellow fluorescent spot. So in the knockdown cells, we see that there's increase of this PLA signal as controls. When the cells are incubated with one or the other antibody, we don't see the signal. And importantly, this is also not due to a redistribution of ZO1 to the cytosol. So it really does seem to be that the lysosomes are in closer proximity to the periphery or to the membrane. So now we see that lysosomes are redistributed to the periphery, and we hypothesize that if they're actually fusing with the membrane, we could detect this fusion through the exposure of a luminal lysosome epitope, or LAMP2, as well as the lysosomal contents in the extracellular space. So what we first did was we performed immunofluorescence for LAMP2 in unpermeabilized cells. And we see this increase in LAMP2 cell surface signal in the knockdown cells. We also confirmed this by using flow cytometry. So again, in unpermeabilized cells, there's an increase in LAMP2 signal in the knockdown cells. And then to detect the lysosomal enzymes that were being released, we collected both the supernatant as well as the cell lysate, and incubated both components with a fluorogenic substrate of beta-hexosaminidase, which is a lysosomal enzyme. And then the ratio shows that there's an increase in the supernatant beta-hex compared to the lysate in the E-level 2 knockdown cells. And then so to turn to a more physiologically relevant model with our collaborators at UCSD and Dr. Wallen's lab, we differentiated iPSC to RPE with E-level 2 knockout. And first, we assessed the morphology of the cells. You can see here in the nice hexagonal shape of the control cells. In the knockout cells, we see that there's loss of this nice hexagonal shape, and we can quantify this by using, again, reshape. There's an increase in the area, increase in the aspect ratio, and an overall loss of this hexagonality score. And on the fatty acid level, there's an accumulation of the substrates of E-level 2 and a depletion of the products of E-level 2. And so because now we have this polarized model of RPE cells in the iPSC RPE, we know that, apically the RPE is in contact with the photoreceptors, and then basolaterally with Bruch's membrane and the choroid. So we wanted to ask whether this, well, first of all, whether this secretion of lysosomal enzymes was occurring in this iPSC RPE model, and secondly, if it was directional. So this time we performed an ELISA for acid sphingomyelinase, or ASM, which is the enzyme that cleaves sphingomyelin to generate to create ceramide. And so first we collected the media from the apical side of the RPE cells in both the nutrient stress, so the B27, and then the full media FBS conditions. We see that there's more ASM being released when the cells are under nutrient stress in the apical side, but there's really no difference between the control and knockout side cells. However, when we collected the basolateral media, again, more ASM being released under nutrient stress, but this increase is really magnified now in the E level 2 knockout cells on the basolateral side. And then to look at this in vivo, we performed cross sections of E level 2 deficient mice, and we stained with LAMP1, so lysosome marker, and we see this redistribution of lysosomes to the basolateral surface in these E level 2 deficient mice. And so then we asked whether we could mitigate some of these effects by supplementing the direct product of E level 2 24/5. And this is also based off of our published work where we found that in aged mice, intravitreal supplementation of 24/5 rescued visual phenotypes on the ERG level, as well as in rod-mediated dark adaptation recovery. And so we wanted to ask whether this supplementation of 24/5 could also be improving RPE health. So the first readout was this unpermeabilized LAMP2 exposure. And so you can see here that after supplementation of 24/5 for 4 days in the cells, there's this decrease or dose-dependent decrease in LAMP2 of cell surface LAMP2. And then to assess lipid asymmetry, we again performed fluorescent Nexin-5 staining. In the E level 2 knockdown cells, there's an increase. And then when we supplement with 24/5, this Nexin-5 and loss of lipid asymmetry is improved. So my summary slide is kind of a model that we're proposing where in young, healthy RPE cells, the lysosomes are functioning and functioning as they should be. However, as aging occurs in epigenetic as well as transcriptional changes, there's loss of membrane integrity, and this loss of membrane integrity and PUFA composition leads to this redistribution of lysosomes to the basolateral surface of the RPE. And so to repair this membrane, the lysosomes are exocytosing their contents, which occurs over time. However, while this plasma membrane repair in the short term achieves its goal of maintaining the cell integrity to avoid death, this constant secretion of lysosomal enzymes and contents may be remodeling the extracellular matrix and contributing to debris and drusen deposits, which we hope to mitigate with supplementation of the product of E level 2 or 24/5. And so this is my acknowledgements, of course, to my mentor, who I did my PhD with, Dr. Dorota Skransker-Kravchik. Thank you so much for your mentorship and, really driving this project and allowing me to work on such an exciting project, and all of the other people involved in both the Science Translational Medicine project as well as this current RPE project. So thank you, and happy to hear thoughts and discuss this further.
Great, thank you very much. Do we have any questions from the audience? I think I see Melanie Gillingham raising her physical hand rather than her
Yes, I am.
Okay.
Yes, I am. Very nice presentation, Emily. Just to clarify, the 24/5 supplementation was intravitreal, not through diet. Is that correct?
Yes, intravitreal.
Okay, and what's the dose that you would do intravitreal of that fatty acid?
So in the mice, we did 0.36 nanomolar, which is very specific. And we also tried other doses, which is in the paper. And it was interesting because half of the dose didn't seem to work, and then double the dose also didn't seem to work. So it was really this kind of Goldilocks, sweet spot.
And what is it? Is it bound to BSA? What's it in?
Yeah, we delivered it with 5% BSA and saline.
Saline. Okay. Thank you very much.
Yeah.
Philip.
Yeah, really cool talk. I was just curious how specific you think this is to lysosomes. Do you think other membrane-bound vesicles might also be getting recruited to the plasma membrane?
Yeah, so I think we got a question about secretory autophagy. And so with the endosomes, exosomes, all of the somes, we focused on the lysosomes because we saw this interesting mechanism of membrane repair, but I would not be surprised if other endosomes or exosomes or other membrane vesicles are also involved in this.
And could you also just give me an idea of the rate at which you think this is occurring? Is this almost like a neuronal synapse? And would that then require like reuptake of plasma membrane to keep the cell from getting too big over time?
Yeah, I think looking at least like the live cell imaging on both for the calcium imaging as well as the lattice light sheet, these events are happening like pretty fast. This like calcium signaling and this like lysosomal movement is happening pretty fast on like the millisecond level.
Yeah.
Yeah.
One
Yeah,
thing,
no, I
one
was
thing
just
maybe
thinking
I
like
can,
FM die or something like that, but it's cool.
well, one of the things which maybe we should say it here, that one of the problems that the cells, E-level 2 cells have, particularly specifically, is to actually produce more membrane.
Oh, okay.
So
Yeah.
because this is where the problem is. So
Yeah.
that's why we think this patching is staying. That's why we can detect it, right?
Mhm.
That's what I
Thanks.
mean. I would just want to say that they probably, I mean, they do seem to be a little bit bigger, but some of them die eventually, right? So it is very uneven. But in general, we think the problem is to produce more membrane instead of and just repair nicely. So the process is stopped or delayed.
Abby, you have a question?
Yeah, fantastic talk, Emily. That was really awesome. I was wondering if you or any other lab members have looked into the contents of these released lysosomes, like if there's increased lipid or even free fatty acid being released from the stress cells that could be contributing to drusen formation.
We would love to do that. Thank you for the question. Yeah. Not only like the released lysosomes, but just the lysosomes like in the cells too. So I mean, there are ways to isolate lysosomes and do like lyso-IP, and I know Dorota's like already, like lipidomics. I want to do lipidomics, metabolomics of the lysosomes. So short answer, no, we haven't done that, but that is definitely something that we do want to do and also look at. Even just like staining for other like lysosomal markers that may be in this like basolateral surface. But yeah, isolating lysosomes from the cells would also be really cool.
Yeah, awesome. Thanks.
Roxana.
Hi. Hi, Emily. That was wonderful presentation. Sorry, I missed the very first part. With all this kind of reshaping of the membrane, I envision that you might have some changes in, at least in a experimental model in a dish, changes in transepithelial resistance. Is that correct? Do you?
Yeah, the interesting thing is we've measured the tier in the iPSC-RPE, and they actually don't seem to be very much changed. And so I think it's because Mhm.
Yeah, go ahead. Sorry.
Because I think because TIR is, measuring the monolayer health, and I think because the cells, and if you see the morphology, let me click back, in the iPSC RPE here, it looks like some of these cells, these smaller cells, are actually being kicked out of the monolayer because they're unhealthy and they're being extruded from the monolayer to really maintain this like monolayer health. And so I'm not super surprised that there isn't like a huge change in the tier. Because I think the cells are either, kicking these unhealthy cells out to maintain the monolayer integrity.
And the follow-up with this cell model, are they challenged with anything? Like in the
So
media, do you have any, or you try to challenge them with something like, performing phagocytosis or,
yeah,
you know,
yeah. So
and
right now, with these, they're the only challenge is that they're not in FBS.
mhm,
6 days. So they have the other nutrients they need, but not, all of the fatty acids that they would like to have. So that's the only challenge right now. But we have thought about additionally challenging like with, phagocytosis,
mhm.
yeah, or oxidative, yeah, the other stresses to the cells.
That sounds great. Thank you.
I'm imagining phagocytosis would feed the cells with some much needed fatty acids. Um, we have a question from Jim. What's up?
So I think you said that it's the mutant cell, the membrane fluidity was changed. It was less, the membrane is less fluid, right? And I was wondering, does that influence would that include the lysosomal membranes? And if so, could that influence the ATP ATP? Could it influence proton pumping into the lysosomes? Have you checked the acidification of the lysosomes? Could that be influenced by this?
Yeah, great question. So the membrane fluidity measurements were done on just plasma membrane fractions. So I can only say that the membrane order or rigidity was increased in the plasma membrane. However, we do think that with the loss of E level 2 and the PUFA production, the other membranes in the cells, including the lysosomal membrane, may be impacted. For lysosomal acidity, we've tried doing, like lysosensor and all of those. We haven't got it to quite work, but we do see transcriptomically that the ATP V0, that acid, that the pump for the lysosome membranes does seem to be down transcriptomically. But yeah, we need to revisit those lysosomal acidity. We have some data showing that the lysosomes do seem to be slightly impacted in their like phagocytosis as well as autophagy functions. So kind of like a not specifically or directly acidity, but kind of a proxy through lysosomal function.
Yeah, but the problem with all of these assays is that none of them were significantly enough to talk about them, you know? It's like everything was just like a little bit. So it's like this biology balancing each other. And I think the fact that they are exocytosing, it's one of basically there is a kind of less available healthy lysosomes or less available lysosomes. That's why we need to bring some more research to it to say, yeah.
But along those lines, why do you think there's less membrane being made? Is that what you're saying before?
Yes. No, because E level 2 is not there, and then DHA is in major component of those membranes. So now we have a slower production of other lipids, and actually we don't see such a huge change in lipid composition except for really very affected so levels of LPSs are higher, LPS, LPC, which means they lack one chain, and then ceramides. So those are really the major things. So we think that the cell is not really working well to produce this plasma membrane phospholipids.
Martin Paul.
Hey, Emily, really very, very nice talk. It's good to have lipid people talking about these things. I mean, for me, listening to your talk, and one point I'm taking home from here is the importance of lipids in retinal function, which for years have been ignored because we are more interested in RNA proteins. So it's good that your team is really working on this. Love the talk. With regards to the earlier question, I think where you were looking at the PS in the plasma membrane, I think Dan was asking, where do you think those dots are coming from? Perhaps if you could localize with Z01 or ZO1, would you be able to tell if that PS is actually on the plasma membrane or is still located within the cytosol associated with mitochondria or ER membrane or something?
Yeah, the tricky thing is that this annexin 5 probe has to be done in live cells, and we've tried like doing live and then fixing and seeing if we could still preserve the signal to do this type of co-localization, and it doesn't seem to work. So it's really in
I see.
these like live, yeah.
Okay, okay. But anyway, that's really a very beautiful work. I know you are interested in 24/5. Do you think that if you provide DHA to these cells, they might be able to do something? I understand because in absence of ELO-VL2, you may not make the 24/5, but will DHA be able to do something about this?
Yeah, we haven't tried just DHA yet with the RPE cells, but I think we would see some improvement in the RPE cells with DHA.
Yeah. And another thing I also find quite interesting is all those lysophospholipids present. Could it be because you don't have the precursor to incorporate into the lysophospholipid to make a complete lipid, or is because in absence of these precursors, you activate a phospholipase, which is cleaving thing? So because it could go in both ways, what do you think
Yeah,
could be?
that's a great point. Yeah. Um, I think so. Yeah, this just gave me I should check the activity of phospholipase to see if it's actually just, the cells need more fatty acids, so they're taking from the phospholipids and cleaving one of
Yeah.
the fatty acids. Or yeah, it could very well be that there's less fatty acids to actually be incorporated, and that's why we only have one. So yeah, that's something to check.
Good job.
Thank you.
Hey, Emily, it's Jason.
Hey, Dr. Miller.
Can you I think you showed the data and I just forgot. But when you went to serum-free media, you were using the neurobasal and B27, right? And that has essential fatty acids to it, right? To allow for synthesis.
Mhm.
So what's the if you don't have the knockout, the ELO2 knockout, what's the overall steady state concentration of ELO2 products in a neurobasal plus B27 situation compared to what you might see if you got stuff supplemented with standard serum amounts? I'm just trying to get a sense of how productive are RPE cultures at making, what you're hypothesizing is important for the membrane. And sort of linked or related to that, at least in primary cultures, we can maintain primary cultures for long periods of time in total serum-free media that has no essential fatty acids. And so I'm still trying to wonder, is that because, like Martin Paul's saying, there's some phospholipases, there's enough reserve somewhere in the cell of essential fatty acids to allow for elongation? Or is this a sort of a phenomenon when you're not challenging RPE, crappy membrane is tolerated, but when you're in an organism with oxidative stress and aging and other things like that, this sort of membrane problem creates cumulative damage over time. That's a big, long set of questions. I'll summarize it. Summary is, in cell culture, cells can live for, at least we can have cells live without a drop in tear for 6 months in serum-free media without any essential fatty acids. So I'm curious, when you do have neurobasal plus B27, how much of the ELV2 products are being made, and whether or not the amount that's made is fine for culture, but perhaps not fine for an organism that is facing more stressful situations.
Mhm. So in terms of, so basically like cells with FBS versus B27 and how much of this like level 2 product is being produced.
That's I mean the first question, yeah.
So all our comparisons are in between B27 for both, right?
Yeah.
So
Yeah. So
because
trying
we
to
always keep, right? So everything is, when we look at the wild type cells, they are in B27 always. So it's the same conditions. So they do manage, they definitely manage working with lower levels of E2, right?
Yeah, because at the moment, the only assay that we've really done in both like FBS and B27 was this ASM secretion. And so we've never, I don't think we've looked at or directly compared the cells in FBS and then B27. Yeah, because normally we just, for both control and the knockout cells, we change it to this B27 condition.
And just as a ballpark, in terms of relative abundance, when you're in the B27 media, how much, let's say C24/5, can you measure in your cells compared to, I don't know, let's say, somewhere between a C20 and a C18 and a C20 product? How much is, just in relative terms, how much is kind of being made?
I think this, oh yeah, I have something,
Got it.
yeah.
This is I
Something like
think
that.
this is what I was looking for. Yeah.
Yeah.
Okay. I'll take a look at that. Thanks.
And I think you had this other question about the impact of aging potentially or other comorbidities. One of the things which we actually kind of argue is that those low lipid conditions are kind of similar to when you have buildups of different deposits between Brooks membrane and RPE, and also subretinal deposits. So basically, those cells are in the kind of like alone conditions. They need to support themselves. So we think that this is all of the above makes those cells really much more fragile to any next stress, right? I don't know, Emily, we thought about it, we discussed it, right? Especially this low lipid condition, that was something we wanted to mimic.
And I mean, the follow-up, this is more just a methodologic thing. I'm really curious if you grow it in not B27, neurobasal plus B27, but like I said, a serum-free kind of standard RPE media, which doesn't contain essential fatty acids. I'm still curious if the cells manage to somehow find a way to make it, because the cells, yeah.
Very interesting. Actually, I'd like to try it.
Yeah.
Jim.
I was just wondering what the photoreceptor phenotype is in the knockout mice.
So structurally, we don't see much like degeneration when we quantify like ONL. But functionally with the ERGs, there's accelerated aging phenotype where in the 12-month-old E-level2 mutant mice, their ERG amplitudes are similar to an 18-month-old wild-type mouse.
Thanks.
Okay, I've been waiting to ask my questions. I won't ask that many. So I'll just do some. The first is really simple. Do you see like a cell growth phenotype where, just if you're plating some ELOVL2 knockdowns or knockouts, they're not repopulating the plate as well? It kind of gets to this general point of can't make membrane quickly enough.
Yes, 100%. Yeah, the cells grow slower.
Okay,
And
yeah.
so originally we were like, oh, maybe they were dying, but then, with all the apoptosis and cell death assays, they're not dying. But yes, they do grow slower, and whenever I seed, I notice that.
Okay, cool. And the other thing is I'm kind of feel like it's very remarkable when you knock out an enzyme that's crucial for lipid synthesis that you're not seeing like gangbusters lipid changes. And some of that could be, some form of biological compensation or uptake from B27 derived fatty acids or replacement of some other fatty acid. But my expectation is always lipid remodeling should be like crazy important. To what extent is that, so I don't have a good way of asking this question. What is the sort of flux of different lipids? How do you know that when you're looking at some kind of lipidome in your knockdown or knockout cells, that's just like you haven't given that change in ELO-VO2 enough time to filter into all the rest of the lipid pools? Or when you are seeing changes, are these the pools that turn over quickly or are smaller pools? Yeah, like how do you think about your lipid data, I guess?
Mhm. I think because DHA is, highly enriched and most, very important, I think that's kind of one of our first readouts that we look at. So not only like the product available to 24/5, but because 24/5 is important for then producing DHA, to really see if there's kind of an impact on the fatty acids, we look at 22/6 first. In terms of other lipid pools and
Can I ask this in a clearer way?
yeah.
Because I just asked a very like long and meandering question. If you knock down ELO-VL2 on day 0 and you do lipidomics on days 5, 10, 15, 20, what are you seeing different lipidomic profiles or the same lipidomic profile on these post knockdown days?
So I don't know if this is going to answer your question, but the reason why we did 6 days is because we were actually tracking this DHA depletion over time in these cultured cells. And it wasn't until the 6th day where we saw that there was this depletion of DHA with E-level 2 knockdown. So I know from day 1 to 6, it's different, but after 6, I don't think we've stretched it out longer than 6 days to see how that changes.
Is that free DHA or like DHA on a lipid? Or both, I guess.
I have to look back and see if it was total fatty acid or free fatty acid. Yeah.
Okay, cool. Awesome work. Thank you.
Thanks.
And does anybody else have any questions? Last chance. Okay.
I mean, Jason, this is more to you though, with regards to your question. When we culture these RPE cells in serum-free media for that long and they are surviving, truly that is not a really physiological condition, what really happened physiologically. So therefore, how relevant are they as a model to model what actually happened in vivo?
Yeah, no, we don't, routinely do this in serum-free media, but I'm saying for a variety of reasons where we were forced to because of, for example, lipidomic analysis where we didn't want contamination from serum. We've done it, and we don't see, they can survive for long periods of time without drops in tear. So I just was mostly curious how much all sorts of mechanisms may come into play to make, even without the fatty acid, may be able to make these things. And I would be really curious to do our basically lipidomic profile in these prolonged serum-free media conditions to see if it's being made. And then, of course, if it's being made, it's got to clearly be activating it from other plasma membrane components that are probably being cleaved and then elongated and then reincorporated back into membrane.
Remember, okay, got you.
Yeah.
Yeah.